In 1876, Koch discovered the bacteria that causes anthrax. He found this bacteria, this odd-shaped rod, in the blood of sheep and goats and cattle who died from anthrax. He noticed it was in every blood sample taken from these diseased animals. Here's Koch. And these are some dead sheep died from anthrax. A dead cow. This is what the anthrax bacillus looks like. It's very unusual in that it is a streptobacillus, that is, it exists in chains. The spores are central, and the ends of the cells are squared instead of rounded. In 1867, Lister published his work on antiseptic surgery, disinfecting instruments with a solution of carbolic acid, which today we know as phenol, very potent disinfectant. He had read the work of Semmelweis and determined that a lot of patients who underwent surgery were dying of infection. They didn't die of the surgery, they died afterwards due to septic infection. And so he advised his staff and his interns to wash all their instruments in carbolic acid. He didn't use chlorine because these instruments were metal and the chlorine tended to make them rust and erode them. So that's why he used phenol instead of bleach. But he noticed that his survival rate of his patients who underwent surgery was much, much higher than those of his competitors, which did not disinfect their instruments. He was also the first person to grow a pure culture of bacteria in 1878. This was the bacteria Streptococcus lactis, and I'll ask you in class where you find this bacteria. You probably have consumed some. In 1879, Albert Neisser, the gentleman we saw earlier in the chapter, discovers the agent that causes gonorrhea, Neisseria gonorrhea. The large pink rounded objects are leukocyte nuclei, and the tiny little specks are the gram-negative diplococci, Neisseria gonorrhea. Here's a close-up. One of the biggest experiments that really rocked the scientific world occurred in 1881. It has a long history that predates the actual experiment, but Pasteur thought that he had discovered a way to make people and animals immune from disease. In actuality, he had. One of his laboratory colleagues, Emil Roux, was studying another disease in animals called chicken cholera. This was devastating the poultry industry of France. They had obtained cultures from contaminated chickens, and Emil was passing these cultures in broth media to keep them alive. They had to be passed every 3 or 4 days, otherwise they would die. Well, they had a big French holiday, and Emil was supposed to come into the lab over the holiday period and pass the cultures, but he forgot to. He was having too good a time. So after the holiday was over, Pasteur instructed him to take some healthy chickens and infect them with the culture that he had supposedly isolated from a chicken with chicken cholera. And he did so. But the chickens didn't get sick. He realized that this culture may be dead. So he started isolating some new agent. Pasteur then instructed him to reinfect the chickens. They just thought it might be an anomaly. To reinfect the chickens, and he did as instructed. The chickens still did not become sick. So now Pasteur's curiosity is really getting the best of him, and he pressures Roux to explain to him exactly what happened. Well, Emil Roux fessed up. He said, I forgot to come in over the holidays and pass the cultures. But I got some new virulent isolate, and that's what I used the second time, but they still didn't become sick. So they thought about this for several days. And lo and behold, Pasteur came to the conclusion that the weak, dead chicken cholera agent that they had originally given the chickens allowed the chickens to develop immunity because it was not the full virulent strain. And so they came up with the first plan to develop vaccines and also to protect humans and animals from disease. So using this model, Pasteur, who was in great competition with Robert Koch, who was German, decided to develop a vaccine against anthrax. So he inoculated 10 sheep with weakened anthrax bacillus to induce immunity like he had seen with the chicken cholera. He waited a couple of weeks, and then he announced to the newspapers, come out and see my grand experiment. I'm going to show you how to make a vaccine and how to protect animals and humans from this awful disease. So all the newspapers came out, and there was a big circus, so to speak. He took these 10 sheep and inoculated them with a virulent strain of anthrax. He also took 10 other sheep that had not been immunized and infected them with the same strain. Within a couple of days, all 20 sheep got sick. And now Pasteur was really sweating bullets because if all these sheep died, he would have been made a fool. Luckily for him, the 10 sheep that had been immunized recovered. The 10 that didn't died. He took organisms from the sheep that recovered, took blood samples, and put them into 10 fresh sheep, or 10 unexposed sheep, and they all developed anthrax and died. So he was the first one to show exactly how immunity can be induced by using a weak or old attenuated strain of bacteria and giving the body this inoculum and waiting produced immunity. There's Pasteur. So he became an instant French hero. He had developed a vaccine for anthrax. He developed a vaccine for chicken cholera, which helped the poultry industry. But his biggest test was yet to come. In 1885, Pasteur was working on a vaccine for rabies. They didn't know what caused rabies. It was not a bacteria because they couldn't isolate any bacteria or see any unusual particles in the blood. But he was working with rabbits, and he was passing this mysterious agent from one rabbit to the next, and he was taking the rabbits after so many days of infection, sacrificing them, taking out the brain tissue and spinal cord, and drying that on plaques of wood to then grind up and try to make a vaccine. Well, lo and behold, there was a little boy named Joseph Meister who got bit by a rabid dog and was in the village where he was working. His mother knew that because her son had been bit by this dog, he was doomed to die. Nobody had survived, not in hundreds of years. If you got bit by a rabid animal, that was a death sentence. So the mother comes pleading to Pasteur, please use your experimental vaccine and save my son. Pasteur was very hesitant because he did not want to be blamed if the child died, that the vaccine didn't work. And he knew he really courted disaster with his sheep experiment in anthrax. But after several days of pleading, Pasteur gave in. And he began to give Joseph Meister the experimental vaccine. And after 2 weeks, Joseph Meister survived. He was the first human in history to survive the bite of a rabid animal. This is the rabies virus. It's bullet-shaped. This is young Joseph Meister. Joseph Meister was so indebted to Louis Pasteur that he later became a custodian in the Pasteur Institute. And there's two stories that whatever happened to Joseph Meister. And I don't know if anybody's been able to verify either one. But in 1940, when the Germans invaded Paris, he defended Pasteur's crypt. The Germans wanted to desecrate Pasteur's crypt because there was always this big rivalry between who was the real father of microbiology, Pasteur or Koch, and they wanted to desecrate his crypt. Well, Joseph Meister would not allow them in, would not give them access to the crypt, and some records say he was shot, others say that they gained access and he went home and committed suicide. Not sure which story is true, but he was a loyal employee of the Pasteur Institute until 1940. In 1882, Koch discovers the bacillus that causes tuberculosis. There's a lung with tuberculosis lesions in the upper lobe. This is a disease that spread from person to person through coughs and aerosols containing the acid-fast rod. And this is a acid-fast stain of the rods. The rods are these the little pink or magenta-shaped filaments that are in the slide. In 1884, Koch used his understanding of bacteria to develop rules in which you could determine if an organism caused a disease or which disease was caused by which organism. And these are known as Koch's postulates. We'll study those in chapter 14. But they're very important. But he was able to establish these rules, and with these rules, many scientists could discover the agents of every single disease that was pretty much known to man. Now, there are some that were exceptions, and so they didn't get every one, but it allowed them to identify numerous diseases and identify specific bacteria that cause those diseases using these postulates. There's Koch studying at his desk and microscope.